In this lecture, I will discuss an alternative visualization method that we have been using in our lab for some time now. The idea is to arrange nodes on a grid based on node similarity or node-node interactions. The idea started over 4 years ago when we faced with the challenge of analyzing a dataset from Ihor Lemishka's lab. The data is a 5-day time series for mouse embryonic stem cells induced to differentiate by the removal of the transcription factor called NANOG. Once embryonic stem cells lose this transcription factor, the cells lose their ability to stay in their pluripotent state. And we talked about it a little bit in the second lecture. So the cell starts to differentiate after the removal of NANOG. Trying to understand the global molecular changes these cells undergo is a major focus of current stem cell biology. So the Lemishka lab collected gene expression using microarray, whole nuclear proteomics using mass spectrometry, histone modifications, and polymerase II binding using a technology called ChIP, which is similar to gene expression microarrays, and is now being replaced by ChIP-seq, which we will describe in details in the lectures that will be presented by Jan Koh. So Ben MacArthur, a former postdoc in our lab, came up with a great idea of how to visualize the changes in expression over time across those multiple layers. The idea was to place each gene on this hexagonal grid by placing the genes that have a similar time series expression next to each other. The hexagonal grid is actually a three-dimensional object called a torus. So when it is flattened to a two-dimensional grid, just keep in mind that the edges roll on themselves and are connected. The key step of creating the grid is a process called simulated annealing. The process is a common algorithm applied in many sciences. The goal is to find the best, or at least a very good arrangement for all the genes on the grid. And in order to achieve this, we need first to have a similarity measure between the variables, as well as a global fitness measure that can tell us how close we are to a good arrangement and potentially the best solution. So the algorithm starts by shuffling pairs of genes randomly and measuring the global fitness. If we get an improvement, we accept the change. If we don't get an improvement, there's still some chance that we will accept a change, and this is computed using the Boltzmann distribution. The idea of accepting changes sometimes that do not improve the global fitness helps us to avoid being stuck at a local minimum. You can read more about simulated annealing online. So the final annealed time series data is then visualized as a movie when the genes stay in their location, but the color of the hexagon changes with time, becoming green if the expression decreases or red if the expression increase as compared to the 0 or control time point. Alexander Lachman, who was a software developer in the lab, implemented the tool GATE in Java as a desktop application. I will now go on and demonstrate to you how the GATE software works. GATE also has an excellent user manual that can help you to get started. So first we're going to try to look for GATE on Google. So you can type GATE Mayan Lab, and then the first link should direct you to the GATE website where you can download the software. And then you click on the download icon on the right, and that depends on your system. The first icon is for Windows and the other icon is for Linux and Mac. The download contains a setup file that you can double click to install GATE, and here you install the software. When GATE starts, you get a registration page. You can skip that, and now you have two options. One is to create and the other one is to load a file, we are just going to use the load option and look at some of the examples provided with the Gate software. This will open a file chooser dialog where you can select from various previous cluster file that we created. We're going to select the nanog file, which is a nanog gene expression changes after nanog knockdown from a previous publication, also by Ihor Lemishka's lab. So once we loaded the nanog file, the Gate software opens, and you can see the hexagonal grid loaded. It is all black because we're looking at the 0 time point. But now we can play the movie and see the gene expression changes over time as waves of activation and inhibitions of various genes. And we see the red genes are genes that increase in expression, and green genes are genes that decrease in expression. We can mouse over the genes to look at the expression level across the time series. And we can also perform gene set enrichment analyses on the sets of genes that are up and down. So if we click on the GMT enrichment button, we can identify the various gene set libraries that we discussed before. One of them is, for example, Go biological process. And then we can select, for example, the up-regulated genes after nanog knockdown at that particular time point. And that provides us with enrichment analysis result. And then we can select and see which genes within the grid are giving us the enrichment for those biological processes that are associated with the upregulated genes after the amount of time that passed since the control time point. So this is it for the Gate demo. There are many other features that the program has that I'm not going to review, but you can try, if you're interested, you can try the user manual, explore the various other features. So the idea of visualizing genes on an hexagonal grid was then extended for visualizing any type of network. So you can think about the genes on the hexagonal grid of Gate as a network of genes that are connected based on gene-gene similarity. And this means that we can have any type of network visualized on a grid. If we go back to our discussion of the sets to networks idea, we can now visualize functional association networks, but we can also transpose those gene set libraries and create networks of terms. For example, we can create a network of all the KEGG pathways based on their gene content similarity. So this is something that we've done for our new tool called Network2Canvas. And with Network2Canvas, you take either a gene set library or a drug set library, and I will discuss a little bit more about this drug set library idea. Basically, it's the same, but now the term is associated with something that is shared among many drugs, and specifically FDA-approved drugs. So if we take a gene set library, we first have to transpose the gene set library, and then we feed it into the sets to networks algorithm to generate a network of terms, for example, a network of pathways. And then we can try to project that network onto this canvas. And for Network2Canvas, we actually created a square grid canvas, not an hexagonal grid canvas. And this is not necessarily better, but is more easily implemented. So once we project the network onto the canvas, we can start anneal the network, trying to position nodes based on their connections or based on their similarity. Once we finish the annealing, we have clusters of nodes that are tightly connected. And to visualize this, we compute the local connectivity of each node by looking at the node neighbors, and then if the neighbors of the nodes are very similar to the center node that we would like to compute the color for, we make the color very bright. However, if the node does not have a lot of neighbors around it, then the color would be dark. So this actually accentuates those clusters that are automatically identified through the annealing process. So once we have those grids of canvases of terms on the grid, we can apply gene set enrichment analysis and visualize the results directly on the canvas. And this is the Network2Canvas application that I would like to demo to you next. But before we go to the demonstration of Network2Canvas, I would like to discuss a little bit about the D3 JavaScript library. So D3 stands for Data-Driven Documents. It's trying to bind data to visual objects, taking advantage of the new implementations of HTML, HTML5 that is supported now by most modern browsers. And by using JavaScript, you can bind data to visual objects using this library. This library was developed by one graduate student from Stanford, and his name is Mike Bostock. The website that documents this has really fantastic data visualization examples and tutorials to really get you started with having the skill of generating those fantastic vector graphics images from your data. So I highly recommend to you to go to this website and try some of the examples on the site and potentially modify them for your own projects. So this is the Network2Canvas user interface. So we can go online and try to briefly demonstrate its functionality. So first we can open a browser and type in Network2Canvas, and this should show you the first or the second link would show you the route to the application. And this is the website. I will make it slightly smaller so it fits in the window. So here you are looking at the KEGG pathway canvas. There are many other canvases that describe those gene set libraries that we also available in Enricher. And you can switch between libraries, depends on the type of enrichment analysis you're interested in performing. So if we look at the KEGG pathways gene set library canvas, you can see that every square on the library is a pathway. So if we mouse over the squares, we can see the pathway name. So each pathway is associated with a set of genes, and some areas are clustered because those pathways have many genes that are shared among them. And this area, for example, is highlighted in clustering. So now you can click on the example, and that populates the text box with the input list of genes that you can potentially enter. And then you can submit the results to get enrichment analysis directly visualized on the canvas. So you have three views. So this is the canvas view that show you the original canvas and the enriched terms highlighted in circles on top of the canvas. You can also look at the enriched terms where only the enriched terms are highlighted, where the rest of the pathways are in black. There is also a network view where the enriched pathways are connected based on their similarity. You can change the colors of the canvas. For example, here I changed it to green. And you can also change the color of the enriched terms on the canvas using the color pickers. Here is the drug set library interface. We created 6 different drug set libraries, and 2 of them are for side effects. One is from the FDA Adverse Event Reporting System, and the other one is from CIDR, which is a computational version of the label inserts. There is also a library for structural elements of the drugs and a library for the ATC codes. Two other libraries are enrichment analysis applied to genes that are differentially expressed after treating human cells with those FDA-approved drugs, and this is data from the connectivity map that I will describe a little bit more in the next lecture. So one thing that we've done, we processed 1 million records from the AERS database and identified 40 severe side effects that are associated with those drugs. And this is the grid that you see on the top left. And then we tried to cross-reference or merge this drug set library with the drug set library for the structural element drug set library that was created by chopping the structure of each drug into pieces that represent chemical structural components, and then trying to apply the enrichment analysis for each list of drugs that are associated with a side effect onto the structural elements canvas. We then rank the side effects based on those side effects that showed the most clustering on the structural elements canvas. And here you can see that cardiac arrest was the side effect that has the highest level of clustering for the structural elements. And this potentially identify relationships between the structural element and the side effect. So for cardiac arrest, there is a strong structural signature that potentially can suggest that drugs with those structural elements would have a propensity to display this side effect. And this is important when designing new drugs, potentially identifying risk factors in the structure of the drug before it is massively produced.